Okay, good morning, everyone. Today I would like to present this article called Selective Single Cell Expansion on a Microfluidic Chip for Studying Heterogeneity of Glioma Stem Cells. This article was recommended to me by Imanol, who found it, and because it's about a similar idea for experiment that we were performing at Rofo Hospital. So the idea is to encapsulate cells, in this case, glioma cells, and here you see the focus architecture to develop the micro droplets. So the idea is to encapsulate just one cell, and it will divide, and we will have these spheres formed originally just from one cell. Here is the architecture of the device. So it has 3 layers. The bottom layer is where the channels that perform this droplet formation are located. Then the middle layer, which has these holes in order to connect the tubing, and the top layer is where the outlets are located, and also the cell inlet and the oil inlet. Here you see the size of this microfluidic device and how they form the droplets using the same oil that we have. And also, here they insert also calcium and EDTA with arginate, so that when the droplets arrive to this oil plus acetic acid, so it's an acid medium, the arginate will gelate, okay? Once that the microdroplets are formed, they are transferred to this Petri dish, and then they will depolymerize the alginate and will recover the aggregates of cells. Okay, so here we have some plots about the diameter of the microdroplets. Here you see the distribution of the size of the microdroplets and also the occupancy, and especially according to the cell density. Here you see the single cell microdroplets, and the idea is that they obtained these spheres in different formats. So they classified manually the formats of the spheres. So they have the tight format is here. There is an image where you can see that these spheres are very joined to each other. Then the loose format, where they are a little bit separated, and the limited size format of the spheres. Then they perform the dead life assay. Here you see in green, with the green fluorescence, the cells that are alive, and with the red fluorescence, the ones that are dead. Also, they perform this test to assess the stemness of the cells. So here you see these markers that are specific for stem cell markers, the NAGOG and also SOX2. So they prove that these are stem cells, and in the different formats, as you can see. Also, they have this question, it is because of the alginate that the cells express genes related to the stem cell or not? Are they affected by the alginate? So they perform the RT-PCR, and then they discovered that the alginate has not an effect on the gene expression. And they discovered this, so that was very impressive. And they also perform another test, for example, invasion, and in the matrigel, they see how the cells invade and the distance that they migrate. So that was another test to prove the stemness of these cells. And yep, so that was basically it. Do you have any questions? I cannot hear. Can you hear me? I cannot hear anyone. Can someone please say something?
Can you hear me now?
Yes, I can hear you, Max.
Okay. Yes, but I can. Let me wait a minute.
Or
Now I can hear you.
yes,
Sorry.
okay.
Sorry, I changed the microphone. Um, have you heard my question?
No, I
Oh,
haven't heard it. Could you
um,
please repeat it?
yes. My question was how they perform the labeling of the cells inside the droplet. They block the droplet and then put the antibodies, or how they do that?
Exactly.
Oh,
They had to depolymerize the alginate so that they can release the aggregate, the cell aggregate. And once the cell aggregate is free, they can um, put the fluorescence. Yeah.
and which is the advantage of using droplets? Uh,
The advantage is to create an aggregate that you are sure that it comes from just one cell.
okay, okay, yeah, good, perfect. Another question? No? Well, who is next? Eric?
Good morning. Can you clearly see my screen?
Yes.
Yes.
Thank you for your confirmation. Well, today I'm going to talk about machine learning algorithms for breast cancer prediction and diagnosis. The main goal of this paper is to predict and diagnose breast cancer using machine learning techniques and find out the most effective based on the performance of each classifier in terms of confusion matrix, accuracy, precision, and sensitivity. Uh, Now we're going to talk about the methodology. We can see here in figure one the proposed architecture. It begins with the data uh, acquisition followed by a preprocessing, which contains four steps, as we see here: data cleaning, select attributes, see target role and feature extraction. Then they prepare data bases used to build a machine learning algorithm that can predict the breast cancer for a new set of measurements. To evaluate the algorithm's performance, show the models the new data for which we have labeled. This usually is done by splitting the label data we have collected into parts. Training set, test sets. Normally, 75 data is used to build the machine learning model. 25 data will be used to assess how well the model works. After test this the model, it was compared the obtained results to select the algorithm that provide the high accuracy. It was identify the most predict algorithm for the detection of breast cancer. Um, Regarding data acquisition, it was used breast cancer Wisconsin diagnosis data set as we see it here in figure two. Feature are computed for a digitized image of breast cancer sample. Breast cancer Wisconsin diagnosis has 569 instances, benign 357, malign 212, and two class, benign and malign, and 11 integrated valued attributes. Regarding results and discussion, after applying machine learning algorithms to the data set, as we mentioned, it was used the matrix, the confusion matrix, accuracy, precision, sensitivity, F1 measure, and AUC curve, as we see in the next page. AUC as performance metrics to evaluate and compare the models and identify the best algorithm for breast cancer prediction. Here, table 1 and figure 3 show the accuracy percentage for the data set. From the results of the training sets and testing sets, we can see here all the classifier has varying accuracy, but support vector machine always has a higher accuracy testing. As we see, 97% than the other classifier. Table 3 present the calculated performance measure of classification model basis on, and here table 2 confusion metrics, precision and sensitivity F measure for two class, I mean benign and malign. Table 2 confusion matrix show that the support vector machine predicts correctly 556 cases out of 579 cases constituted of 201 malign cases that are actually malign 356 benign cases that are actually benign, and 11 cases incorrectly predicted, including 11 cases of malign class predicted as a benign, and one case of benign class predicted as a malign. From the results of table 3, we can see the percentage of precisions, 98%, sensitivity 94, and F measure 96 of support vector machine are higher than the other classifiers. Then we can see here in figure 4 the ROC curves of each machine learning algorithms. ROC curves is an important metric for the performance of classifier. Here, the area under the ROC curves is calculated. The area, I mean, the area is bigger, the performance of the classifier is better. Here, the support vector machine has the highest AUC score, 96, while the AUC score of decision free, 94%, is the lowest shown in this table 4. As a final conclusion, we can say that the support vector machine has demonstrated that its efficiency in breast cancer prediction and diagnosis. It achieved the best performance in terms of accuracy and precision. So that's all from me. Many thanks for your attention.
Thank you. Any questions?
Eric, I have a question for you. You mentioned that the support vector machine was the one that was the best. Are these like three different algorithms that or three different tools, the support vector machine, and the other ones? Are these three different algorithms that they perform, or are they plugins or tools?
I'm sorry, can you repeat the question, please?
Yes. You mentioned these support vector machine and other random forest. Are these different plugins?
Five algorithms, 5 kinds of algorithms. Yeah, support vector machine, decision tree. Yeah.
Okay.
Go ahead.
Yeah, I wanted to know that if they were different algorithms that they developed, or that these are tools that are already created by someone else, for example.
I don't understand exactly your question. You mean if the results has different? I don't understand your question.
No, if the algorithms were made for them, or they are libraries that are
Okay.
for everyone.
Okay. I got it. Okay. In this paper, they use software the Python, but they use library. The library, the call is SQLearn. It is available to show and to calculate these all parameters with different algorithms.
I see. Okay. Thank you very much, Eric.
Good. Any other question? Okay. Who is next? You can look in the table I sent for in the email.
Juan, could you please continue?
Okay. Can you all see my screen?
Yeah, but can you zoom out a bit? Yeah, zoom in. Good.
Okay. Today I'm going to talk about microparticles of hydrogel and its ability to the cell adhesion. So in this study, they use a polymeric chain that contains benzophenone molecules that was synthesized and coated on bare organic substrates, and then they use irradiation using UV light to photoactive this molecule of benzophenone and enable the formation of this network to attach in these organic substrates and later with the cells that they want to attach. So they found in their results that from immune staining and cell addition experiments that at a certain surface density of active benzophenone play a role to the bioactive properties of laminin decorated spots, while using the FTIR results indicated that laminin, that this protein, was stable upon the irradiation using UV light up to 18 joules per centimeter square. So the use of these particles suggests that in many cases the interactive and biomimetic surfaces are favorable to prevent such foreign body responses and enhance the biomaterials performance, and because the implanted materials should not only provide the structural support for damaged tissues, but also interact with these tissues and ideally promote their regeneration. So they say that an ideal scaffold surface provides a microenvironment in which the receptor-mediated cellular response are supported, while non-specific cell adhesions and interactions are effectively prevented. So in this work, they present a highly efficient and versatile strategy for the generation of dual functional with this polymer PDMAA based hydrogel coatings on bare standard cell culture substrates using this CH Cheek reactions. Through these Cheek reactions and the benzophenone functionalities are allowed to covalent cross-link these polymer chains and covalent this immobilization of this polymer network on bare cells culture substrates, and the functionalization of the partially cross-linked biopassive with this polymer network with the bioactive molecules such as laminin or some other protein. In this case, they use, I think, integrins. And it could be done simultaneously, this process, with a simple and fast UV irradiation step. But it is known that too high energy dose of this UV light irradiation may degrade or denaturate organic substrates, including proteins. So too high energy dose may also cause side reactions that can cross-link proteins to their organic molecules without the need of these photocross linkers. In the results for the strategy of this surface modification, with this DMAA, this polymer that was the main component of this copolymer, serves as a hydrophilic matrix to prevent non-specific interactions with biological environment. And the second component was this another polymer, MABP, that provides the photoreactive functionality. They use ethanol to dip or spin coated on bare organic substrates like PMAA or PS, and later establish a covalent bond to connect the two polymer chains. In this illustration, we can see the process, the whole process. We have two coatings with UV to generate first the interactions, and then to adhere or to cover to drop coating the laminin, these integrins, to later culture with cells and later adhere to these cells, and they perform these assays. So to the controlled grafting of laminin, they use 5 surfaces with different surface densities of unreacted benzophenone. They identified with the A, B, C, D, and E. The laminin molecules on the surface were exposed to UV light irradiation using 3 different energy doses. And we can see in these images the results of these additions using the different dose of UV light. And they conclude that, as we can see in the F image, that the irradiation was too high, and it was produced a decrease on the proliferation of the cells in the surface. So they show in this another illustration these assays performed during 9 days to different surface density of this active benzophenone to cross-link the protein laminin, and the energy dose to cross-link the polymer PDMA and the laminin. And they found that like I said, 18 joules per centimeter square, they found the growth or and the proliferation of the cells were efficient, because they found that using these micrographs that the cells the cell's morphology was flat on bare PES surface and laminin degrade PDMA, indicated they call happy cells, so they can proliferate along the polymer. But when they are full, they change the morphology, and they say these cells are unhappy. And when the surface density of these cells is high, the cells crowding and rounding occurs, this morphology, indicating these unhappy cells, which leads to cell detachment from the surface. So they found finally that a good attachment occurs till 7 days, and no bigger energy dose than 18 joules. And yes, that's it. Thank you.
Okay, any question?
I have a question. Did they try to see if it was reusable, or just did one experiment with each?
Good question. I cannot say it, and I think they didn't show this. I think it's one experiment. I don't know. They didn't say that is reusable substrate or reusable polymer. Sorry, microparticles of hydrogel.
Okay, thanks.
It is worth to mention that this article is from a group we are collaborating with. So if you have any particular question, we can ask directly to the authors, who will respond us. Okay, who is next?
I'm next. Okay, so I'm gonna be presenting this paper, which is an stimuli-responsive luminescent nanomaterial which is based on lanthanide modified carbon dots. And they are using here for multilevel information encryption. So the stimulus-responsive fluorescent materials are showing great applications in potential information storage, security, or anti-counterfeiting due to their controllable optical properties under the external stimuli. So stimulus response lanthanide anti-counterfeiting materials have become a research hotspot in recent years, but the problem is that most of them only demonstrate single stimulus responses. So in this paper, they're gonna be using carbon dots, which are carbon nanoparticles that are less than 10 nanometers in size. And they are typical luminescence materials that can absorb various excitation light and convert it into visible light emission. It also has many advantages involving controllable luminescent color, outstanding light stability, low toxicity, and biocompatibility. So in this work, the authors fabricate carbon dots with europium. So the first thing they did, they synthesize them and then characterize both carbon dots with and without europium. And one of the things they saw was that the average particle sizes were about 2 and around 4 nanometers, respectively. So next, they evaluated the fluorescence spectra obtained of the europium nanocomposite, and they saw that the optimal emission peak was located around 525 nanometers, and they saw a strong green light emission that could be observed under a UV lamp. So the fluorescence spectra of these nanocomposites, they tried it in different pH buffer solutions, and they saw that the fluorescence intensity at 525 nanometers was significantly enhanced with the increase of pH from 1 to 9. And the fluorescence color of this nanocomposite solution also switched from colorless to green under UV light radiation. And they also saw that when the pH goes down, they also move from the colorless they move from colorless to green when the pH increase. So the fluorescence intensity of the nanocomposite is enhanced when you increase the pH of the solution, and it's reduced when the pH decreases back to 1. And this sensing process can be reversible for at least 5 cycles, and indicated that this is a promising reversible pH sensor. They also saw that this carbon dots exhibited monodispersity under pH 6, while aggregation occurred when the pH decreased to 1. So these results indicate that the pH responsive fluorescence signal is originating from the aggregation and disaggregation of these carbon dots under different pH conditions. So they also tried what happened when they used different molecules, and they saw that, for example, with the increased concentration of DPA, there's a transition from green to red, and with the increase of presence of copper ions, there is a trend to turn from red to green. So as a test, they use this invisible fluorescent ink at pH 1, and they wrote in Mohs code the word for computer with this invisible fluorescent ink, and the information was invisible under both natural light and UV lights. The fluorescence color of the word computer then turned green when they encounter TEA steam. Then the information can be clearly seen as here in C. And due to the prominent pH reversibility of these carbon dots, the information can be then re-encrypted back into a blank paper by placing a HCl vapor after reading it. So since these outstanding pH reversibility and PDA responses of these nanocomposites, they wanted to test and see they synthesize if this was also applicable to anti-counterfeiting. And they saw that when the invisible ink with a pH of 1 was coated on a pattern, the information was invisible under both sunlight and UV light. After treatment with TA vapor, the pattern was still invisible under sunlight, but can be seen with a UV light. So in addition, thanks to this sensitive effect of the lanthanide europium by DPA, they also obtain a multicolor emission pattern, and the pattern consists in two parts: commercial green ink and a green fluorescent ink made with the composite at a pH of 9. And the pattern can't be seen under natural light, but an overall green pattern can be seen with a UV light. And after spraying DPA, you can see here, I don't know if you see my cursor, the color of the birds that were drawn with the nanocomposite, they turn red, while those that have been done with the green commercial ink, they remain unchanged. And these patterns can also return to the original green color after spraying them with a copper solution. So in conclusion, they use a fascinating luminescence phenomena that indicated that the luminescence of anti-counterfeiting labels can be made with these carbon dots and European nanocomposites, and they can be modulated by acid-base, DPA, and copper stimulation, thus increasing the difficulty of forging. So that is all. Thank you. Any questions?
Yes. What are they look interesting, but what are the potential applications they mention for this tool?
They say they want to apply it basically to encrypt information, but they also said it can be using for counterfeit. For example, when you have I don't know if it's just like applying a water thing. I don't know exactly remember the name in English. Una marca de base de agua.
Watermark. Yeah.
A watermark.
Yeah.
So that you can avoid counterfeiting stuff.
Okay. Yeah, I imagine for toys, for the chemical toys for the kids could be useful, but maybe there's another more commercial application. And have they tried it? Because you show solutions, but have they tried it at molecular level? How many dots do you need to see that effect?
It doesn't take that much though, because the dots are luminescent. I mean, it's usually a solution, but it doesn't have to be like quite high.
Yeah, but
The
you are
concentration.
looking the whole solution. But if you go
If you go at the molecular
minutes,
level, the same happens in all of them. If you have one and you add the things,
but
it's going to be the same.
it not depends on the intensity of the emission of the dots. Can you see the same effect if you have just one dot,
Yes,
or okay, you sure?
I think so.
Okay. Well, another question. Okay, who is next? Look, you can look in the table I sent by email, and we don't have to wait.
I have looked in the table, but there are no more presenters.
Yeah, it was Luis. After Rocio is Luis. Luis, do you have something to present?
Hi, I, I, I can't, I couldn't prepare for this week because the present week is the project, and I couldn't to prepare because the 9th of March is the deadline to a project for the lab. and if you can please I will to expose my paper on the next week
Okay.
of the
But
the
next,
Paperthon.
okay, I understand. But next time, send me an email with time so I can tell to other of the members of the group to present. The idea is to have 7 presentations each week, and each presentation should be 5 minutes.
Okay.
Maybe another minute of questions, but today some presentations last longer, but since we are less than 5 people this week, it was not a problem. But for the next week, remember 5 minutes for presentations. Just focus in the graphs, explaining the results and the conclusion and how it works, and that's it.
Okay.
Well, another thing, yes, don't forget with time also to add the let me wait, I will stop the recording.